Good day, everyone, and welcome. I'm Jack Van Horn from the University of Virginia, and I'm very proud to welcome you to this edition of our 2022-2023 Foundations of Biomedical Data Science Seminar Series. This lecture series is supported by UVA's Integrated Translational Health Institute of Virginia, the UVA Brain Institute, the College of Arts and Sciences, the School of Data Science, and through a grant from the National Institutes of General Medical Sciences. Since 2016, the Foundations of Biomedical Data Science Seminar Series has focused on the use of data science methodologies on artificial intelligence, deep learning, and statistical modeling as they pertain to differing themes in the biomedical and health sciences. This year's theme is data science and the public health consequences of the COVID-19 pandemic, where each Friday we enjoy presentations from leading thinkers about the issues, the promise, the opportunities, and the hurdles associated with understanding the secondary health effects of COVID-19. These include delayed health checkups and treatments, mental health concerns, drug abuse, increased obesity, and the long lingering health effects associated with the SARS-CoV-19 virus. Participants selected for our Biomedical Data Science Innovation Lab program will leverage these presentations as vital material for our culminating in-person grant project development workshop to be held at the Lodge at St. Edwards Park in Seattle, Washington in June of 2023. Today, I am delighted to welcome our speaker, Dr. Shweta Bansal from the Department of Biology at Georgetown University. Shweta is a Provost Distinguished Associate Professor of Biology at Georgetown University and trained as a network scientist and disease ecologist from the University of Texas at Austin, and was a fellow of the prestigious Rapid Postdoctoral Program of the U.S. National Institutes of Health and Department of Homeland Security. She earned her PhD from the University of Texas at Austin, her master's degree also from University of Texas, and her bachelor's of science degree from Santa Clara University. At Georgetown, she leads an interdisciplinary research group which specializes in big data-driven mathematical models to address how social behavior and spatial dynamics shape infectious disease transmission and how knowledge of such processes can improve disease surveillance and control human and animal disease systems. In continuing with our 2022-2023 Biomedical Data Science Seminar Series theme, today's lecture is entitled Infectious Disease Surveillance and Modeling Through Spatial Big Data. Shweta will remind us that during one of epidemiology's formative moments, the noted John Snow mapped the London homes of people who had cholera and succeeded in highlighting the risk of diseases associated with the Broad Street pump. Since that time, spatial investigations have played a critical role in improving our understanding and the associations of risk between risks and disease outcomes. Modern electronic resources help us to carry out spatial epidemiological studies through large-scale digital health and provide opportunities by increasing accessibility to populations over space and time with data on personal beliefs, behaviors, and health outcomes in an unprecedented breadth and depth. In this talk, Shweta will be discussing case studies where spatial big data has improved the ability to model and describe ongoing challenges, such as spatial big data, or where big data has become more pervasive in informing disease surveillance, disease control, and public health policy. So as always, we are streaming this lecture live and for recording via YouTube. And if you're watching on YouTube, thank you so much for joining us. Also, our specially selected 2022-2023 Biomedical Data Science Innovation Lab participants and alumni are encouraged to submit any questions they have via the chat feature in their Zoom sessions. I will synthesize these questions and ask them on your behalf during the last 10 minutes or so of the lecture. And with that, welcome, Shweta. We are really looking forward to hearing your presentation today.
Thank you so much, Jack, for the kind introduction. And I am thrilled to be here. Good afternoon, everyone who has joined. At least it's my afternoon, maybe it's morning where you are. So just a good day to you all. And yeah, really thrilled to be joining everybody. I will try to run through the material I have, but really I'm excited about discussion at the end with you all. So really what I want to just kind of jump right in here and bring up social behavior in the context of infectious disease. This is really what we focus on, and really social behavior is kind of fundamental to infectious disease, and that's really probably not a surprise to anyone joining today after the last 3 years that we've had, right? So interactions at the individual scale between each of us, right, are kind of central to the disease transmission process, from sexually transmitted diseases being relevant to STI transmission, to handshakes and face-to-face conversations being relevant for spreading respiratory pathogens. We really care about these interactions that are kind of baked into our society. But beyond the individual level, right, kind of zooming out from there, the interactions that happen between populations through air travel, when we move from one location to another, or via livestock movement or wildlife translocation, when animals move from one location to another, those types of interactions really can help diffuse pathogens over large geographic scales. So really, we care in the work that I do, we really care about both of these types of interactions sort of across different scales. But of course, we also want to recognize that interactions are not static, right? This connectivity within populations and between populations isn't static. It gets disrupted by preventative behaviors that we take as hosts, as well as public health policies that get put into place to really prevent, to suppress disease transmission. There's also, of course, the disease itself that can have an impact on our social behavior by kind of keeping us home when we're not feeling well, or really leading us to kind of avoid each other when there are symptoms that are present in the population. So really thinking of this as a classic sort of complex system that has interconnectedness, nonlinearity, and feedback loops is really kind of the big picture I keep in mind. And what I'll contend is that really that host behavior at the moment is the single biggest gap in our understanding when it comes to thinking about infectious disease spread. Because we don't understand behavior at a fine scale, we can't really predict disease emergence well, we can't forecast diseases well, and we really can't get ahead of, and be able to eliminate diseases before they take hold in populations, which has, of course, become painfully evident over the last 3 years. But fortunately, this is really the focus of my group's work. It's convenient that I'm making this case that this is really important, that this is really what we focus on. So we do this work in human and non-human animal systems, and all of this is really fueled by a group of fantastic early career scientists, all the way from undergrad to senior scientist level, and I'm excited to kind of share some of their work with you today. So for the purposes of today, I'll of course just focus on the human side of things, just to keep things a little streamlined. But if anyone has questions about the more animals work that we do, I'm happy to say more. So in the past, the human side of our work really has focused on sort of the individual level. Right here, we really try to understand how contact between individuals, and I'll explain more of that in a minute, drive disease emergence, spread, suppression, and how disease itself impacts behavior. But behavior, like I was just saying, doesn't just matter at the individual level. It also matters at larger scale. So our recent work is really also focused on behavior at a landscape scale, basically, where we look, kind of what happens when we zoom out from individual behaviors and look at how a population of individuals is responding collectively to a disease. We focus on kind of how the social structure kind of leads to structure across geography that gets created by communities interacting with each other, and how this drives disease emergence and diffusion, and of course how we can do disease surveillance better and control better, more optimally. Now, we really don't have to look very far, I would say, to have motivation to care about disease at large scales, to care about these landscape effects of disease transmission. The COVID-19 crisis has kind of been a textbook example of what we call a patchwork pandemic, right? And these kind of, I've got an image here up of the two largest democracies in the world, which take a very kind of decentralized approach to doing pandemic surveillance and response, to kind of highlight what I'm talking about here, right? So in a patchwork pandemic, really, there's not only variation in where cases occur and when they occur, but that there's also geographic structure to that variation. And it's not just randomly distributed around the country. And that geographic variation is due to a whole host of factors: demographic, environmental, economic, in terms of systemic inequities, public health resources, how those are distributed, right? A whole host of factors affects this. And then all of that variation at a geographic level gets kind of amplified because we are not living in islands in these communities. US counties, for example, are not islands. We're all connected to each other via physical mobility, individuals moving back and forth between locations, but also, of course, in the 21st century, social connectivity through social media and other information sources. So really what this connectivity does is it, kind of amplifies the effect of that geographic variation. And really, there's, from a public health perspective, there's a lot of dangers of being in a patchwork public health situation. It, results in public health surveillance and response resources kind of being spread too thin, always kind of being behind the curve. And it's also a situation when, in terms of research, there's always kind of a mismatch between the experience and reality at the local level versus kind of what is being decision-making processes at a national level. So really communication becomes a really complex thing in such a patchwork situation. And of course, right, beyond the public health perspective and all the complexities that brings, from an epidemiological perspective, right, understanding what transmission potential is in a patchwork gets really quite messy as well, right? So transmission potential in each community at a given time really depends on, of course, the susceptibility of that location, the transmission factors in that location, but also how that location is connected to other locations. And each of these factors, each of these components really is driven by a variety of behavioral and other factors, which in turn are driven by kind of a complex socioeconomic, political, demographic, and environmental landscape, which all has its own geographic variation. So really what we'd love to be able to do in the future is have a big mechanistic understanding of each of these processes and then be able to integrate all of them to be able to predict what transmission risk looks like across space, across a big country, like a big heterogeneous country like the US. But at the moment, we're not quite there. Right? And there's just a ton of data that could really solve this problem because we're really lacking a mechanistic understanding at the moment of how to deal with this. And when it comes to data for understanding geographic structure when it comes to infectious disease spread, right? There's a lot of data challenges that we kind of still face, right? Data is collected in lots of traditional settings through federal sources, for example, and state sources, but there's, varying data definitions that they use, varying reports, kind of reporting mechanisms. There's a lot of delays that can happen. Deduplication, for example, is challenging. So what we'd ideally love are data sources that are, high resolution, large scope, high coverage. There's maybe a uniform data collection method, and that if there are biases in the data collection method, that they're known, right? In the past, this has not been the norm. So, in the US, we've tended to actually be in a sort of pretty data poor setting, despite the fact that we're a high resource public health setting. And so before the pandemic, and honestly, till this day, for non-COVID situations, right? Disease surveillance kind of looks like this. It's at maybe the state level in the US, and it's really at a qualitative scale, right? Where we might know that disease is high or low or minimal, right? But it's not particularly detailed. So in comparison, kind of what we've had with over the last 3 years has kind of been a sea change in fine-grained disease surveillance, thanks to states reporting a lot more rapidly, and thanks to heroic work, honestly, by journalists and academics, where not only scientists and policymakers have become really used to kind of fine-grained county-level maps of disease outcomes, but just the general public has as well. Now, this is, in terms of like measuring where disease is in the country, but in terms of where behaviors are occurring that increase or decrease risk of infection, right? Behavioral surveillance is still quite poor. And so this is where, we can really kind of think outside the box and try to think about non-traditional big data sources, which are, by the way, not collected for epidemiological purposes, but can maybe sort of provide an opportunistic, provide an opportunity for us to like repurpose their use for scientific and health decision-making purposes. And really the past 3 years, again, has sort of been a landscape shift, a sea change in kind of the unprecedented number of open data sets that have been created and have also been shared with the scientific community, giving us a chance to kind of explore how using these data might work for thinking about disease transmission. And such data sources really kind of open up a lot of opportunities for us, right? They come from, they can come from administrative data sources, social media data sources, mobile records, right? These, again, are just data that are just being collected passively for other purposes. And the question is, what does it look like if we can repurpose these for scientific and epidemiological purposes? And these really provide a lot of opportunities, right? There's kind of sort of improved resolution, timeliness to it, maybe access to certain populations that are hard to collect data on when it comes to traditional data sources. There's a whole host of challenges that come with working with these data as well, right? Representativeness continues to be an issue because there's not an underlying statistical study design here. You kind of get what the data source has collected, and I'll talk more about that. There can be issues of noise, issues of scale, issues where maybe traditional data is collected at a different scale than non-traditional data, and so integrating these becomes a challenge. And then, of course, there's a whole host of sort of ethical and privacy issues that the field is still really grappling with, and I'd say we're nowhere near having these resolved, but I think continuing to talk about them is important. So as Jack said, what I'd love to do today is kind of share some of the stories from our work where we've kind of tried to use some of these novel data sources to think about behavior sort of at that landscape scale, and then also talk about how we can think about these data sources to think about where disease is and where other health behaviors are or health outcomes are at landscape scales. What we really aim to do with these data sets is do mechanistic modeling, and I can explain what I mean by that during the discussion. But what I'll focus on today, since the focus of this group is kind of biomedical data science, I'll focus on the more statistical modeling of these big data sources and kind of talk about how we use them. And so before I go further, I kind of want to roll the credits here for the junior scientists involved with this work that I'll describe today, and I'll give kind of a visual shout out to the ones that led each project as I go through. So I'll start, like I said, on the behavior side, kind of using some big data sources to think about behavior and some of the challenges that kind of come up along the way. So the first is kind of what I want to talk about is contact. So really contact is the most fundamental behavior to disease transmission. And it's what it's defined as is the social interaction between two individuals that is capable of transmitting infection, right? So again, if you're talking about a sexually transmitted disease, this might be a sexual interaction or something like needle sharing, which does fluid transfer and allows the disease to transmit. Whereas for respiratory infection like influenza or SARS-CoV-2, contact can be a conversation in close proximity or even just being in the same room and sharing the same air. And what might be surprising to those of you who are not in this field, right, is that even though this behavior is so fundamental to our understanding of disease transmission, we don't understand it very well at all, right? We have small studies that give us static snapshots of what this behavior looks like in individual populations, and we really don't have a sense of, geographically, how does this vary? How does it vary over time? How do people respond to disease? And certainly we have no idea what this looks like in the US, because most of those studies I mentioned are done outside the US. So to better understand this, what we did is we, worked with a survey that's been a large-scale survey that's been running since April 2020, that receives about 50,000 responses, or received while it was running 50,000 responses every single day. So that about in the first year, they had about 18 million responses. And one of the questions on the survey basically asks people in the last 24 hours, how many contacts have you had? And so we started with this data and we modeled it using GAM models, if you're familiar with those, generalized additive models. And really what we were trying to do is characterize the spatial distribution of the number of contacts of these 18 million people distributed around the country, which really gives us a measure of kind of how each county in the US, in this case, how each community is on average at risk for infection rather, right? As you can see from this map, which is kind of the result of our study, there's a ton of spatial variation, a ton of geographic variation in the country. And if you look carefully, you'll notice that, sort of more urban locations like there are on the coast, but even in states like Georgia, right, the location where Atlanta is, that urban area around Atlanta, has much lower contact, so much lower risk of infection than more rural parts of the country. So being able to, again, understand this at this large scale is kind of critical to being able to think about risk for infection. But as we thought more about this data, really what we started wondering was what sort of biases might exist in this data, right? The survey that I'm talking about, the CTIS survey, was administered through social media, and so it isn't statistically designed to be representative. And on top of that, the survey has been running kind of through a very political, highly polarizing time in our country. So we figured that there would be a reasonable selection bias in who was, likely to respond to the survey, and that, really there would be sort of social desirability maybe at play as well. And so really to estimate whether this was the case, we would need ground truth data for something that the survey measures that we could compare against, right? And so in this context, we turn to thinking about vaccination, because vaccination is something that we have sort of ground truth data for. We have official records, which turns out to be complicated in itself, but we do have official records on, for each county, what percentage of individuals are vaccinated. And so we can use the survey data on people reporting whether they've been vaccinated or not, and to compare against this official data. And by comparing this data to the survey data on vaccination, what we were able to confirm is that the survey, which is the orange dots here on these plots, these happen to be for two sample locations, Omaha, Nebraska, and San Francisco, California. And we noticed that across locations, right, the orange dots, which are the survey data, versus the blue, which is the ground truth sort of official record of vaccination, the survey really seems to overestimate vaccination behavior. So by sort of modeling what that difference between the orange and the blue is here, we were really able to come up with a single kind of bias estimate for each US county, so that we could then use that in future uses of this data. And that came soon after, when we were trying to kind of use this survey again with its millions and millions of responses to characterize masking. Now, masking is, like contact, is kind of notoriously difficult to quantify at a large scale. And 3 years into the pandemic, or 2 years when we did the study, right, we still really only had a core scale idea of what masking looked like on really short-term estimates. And so really this made studies where we were trying to understand national risk for individual communities really, really challenging to do. So we went back to the CTIS survey and looked at responses to a question about masking, and we used sort of a Bayesian approach here to deal with locations that might have small sample sizes. And we included that estimate of bias that I just talked about, that we produced using the vaccination data. And what this model allowed us to do was it gave us an ability to estimate masking at a fine scale, so at the US county scale, monthly after bias correction, and be able to have much more realistic estimates of what masking looked like around the country. Because of course, if you looked at the raw data, it would show that 90% of every single county in the US, people were masking in, and that we know is not true from our personal experiences. So really having much better realistic estimates of what masking looked like is, gives us the ability to make much better estimates going forward in terms of infection risk for both COVID, but then other respiratory infections as well. But really what we, one thing that kind of kept bugging us in the back of our minds is that we really didn't see, as surges of COVID happened in the first 2 years especially, we really didn't see that mapping to associating with behavioral changes that might be happening in the population. So to give an example, if we look at the winter 2020 surge of COVID in Chicago, right, the red curve here is cases, and the sort of dashed ones here are different measurements of behavior that we expect to be associated with disease. And if we looked at, things like masking and contact and number of people staying home, right, most of those seemed pretty flat. They seemed to change once cases went up enough, but really it was a response to cases going up. But what we didn't see is an increase in, worsening in these behaviors that would explain why the surge started when it did. And so really what we started wondering is, are we measuring the right thing when we measure contact and masking and stuff? And really, what are the risk factors when it comes to respiratory disease transmission? So what we started wondering about was like what the context is in that a given contact happens in. Just because I have an interaction doesn't necessarily mean it's a high risk interaction for a given respiratory disease, right? And so it turns out if you think about respiratory disease, and especially SARS-CoV-2, it's an airborne infection, which seems to transmit a whole lot better in indoor environments where ventilation tends to be poor. So we realized what we really needed to get a handle on was what indoor behavior looked like. But indoor behavior is, again, one of those things that, despite its importance for big pathogens like SARS-CoV-2 and influenza and pertussis, really, we don't have an idea of how much of our time in the US or any other place is spent indoors. There's a EPA study from the early 90s that tells us something like 87% of all of our time is spent indoors, but it's like one value for the entire US, and it's really limited on kind of what it tells us. So here again, we kind of tried to use a novel data source, and this time it was a mobile phone app-based location tracking data set, which really kind of gives us a handle on, of 5.5 million locations around the country where people are spending their time, where people are visiting in a given day. So what we did is we took these 5.5 million locations and we classified them as indoors or outdoors. So examples like schools and office buildings and, supermarkets, grocery stores were classified as indoors, obviously, and playgrounds and amusement parks and farmers markets and cemeteries were classified as outdoors. And so what we did is we took all this data, we split it up by indoor versus outdoor location. And then we aggregated it again to the county level to figure out for each county and each week, what is the tendency of human visits, activity, to happen more indoor relative to outdoor. So we get this type of results from that, where what we're measuring here is just across time. So this is a sample year of 2019, so before the pandemic, to just have an idea of baseline. And the y-axis here is that indoor activity, right? So if this is a value above 1, then people are spending more of their time indoors than outdoors, or more to indoor locations than outdoor locations, and vice versa if the value is below 1. And so we can just look at these couple of locations. So Suffolk County, where Boston's located, right, seems to have a peak in indoor activity when it's cold outside, whereas people go more to outdoor locations when it's nice, comfortable in the summer, right? Where somewhere like Maricopa County, which is where Phoenix is located in Arizona, right? You do see a peak in indoor activity in the winters, but then you also see it in the summers, where in Phoenix, it's pretty miserable to be outdoors, right? And so clearly so we got one of these curves for each county in the US, all 3,000 or so counties. And we thought, okay, there's a lot of heterogeneity here. There's a lot of variation. So let's try to figure out if there's actually any geographic structure to this heterogeneity. And so what we did is we just essentially did a simple kind of time series clustering approach, took all these time series and tried to cluster them based on their shapes. And this is kind of the results that we got, right? So basically, all the locations in the country kind of fall into a group. So most locations are either falling into this light blue or dark blue group, where the light blue group is kind of looks like the Boston curves I showed you, right? Where indoor activity really peaks in winters and it's pretty mild in summers. Whereas the dark blue locations are more like Phoenix, where there's peaks in winter and summer when it comes to indoor activity. So this is, kind of really important to us from a disease risk perspective to know kind of which locations are at risk at different times of the year, and really is important to being able to predict seasonality and behavior. And of course, beyond infection spread, right, climate change adaptation is really going to kind of affect what this map looks like and how we're going to be able to cope in different parts of the country. The last example I want to talk about on the behavioral side is a little bit more mechanistic, right? So it's of course important to kind of develop these maps to kind of understand the geographical distribution of these behaviors, to understand the geographical distribution of risk. But sometimes we want to dig a little bit more and figure out what's causing these behaviors. So the example I want to go to here is vaccine refusal behavior, which, if you want to talk about consequences of COVID, the pandemic, this is going to be one of the huge consequences that we're going to, kind of feel the effects of for a long time. So vaccine refusal, just to define it, right, is the intentional refusal of vaccination despite having access to it. And this is often for philosophical or religious or sort of personal rights reasons that individuals refuse vaccines even though they have access to it. And so this is pre-pandemic work where we're just looking at the distribution of vaccine refusal for childhood vaccines. So these are parents refusing vaccines on behalf of their kids, things like MMR and pertussis vaccine. And really, what we notice here is that there's clusters of high vaccine refusal and low vaccine refusal locations that turn up in the country. What I mean by that is locations where vaccine refusal is high are surrounded by other locations where vaccine refusal is high. And disease epidemiology kind of tells us that these clusters are a much bigger threat to us rather than if vaccine refusal behavior was kind of just distributed randomly across the country. Because there's these clusters and because humans tend to interact more with those that live in nearby locations to them, they are more of a sort of a hotspot for disease emergence for diseases like measles. And so it's really important to understand why this sort of clustering occurs. And so we had a couple of different hypotheses here, right? We had a hypothesis that maybe these clusters occur because there's some underlying socioeconomic traits of individuals that make them more likely to refuse vaccines. So one of the common known ones is that if you're college educated, you're more likely to refuse vaccines, or if you're high income, you're more likely to refuse vaccines. And because income and education tend to be geographically clustered, then we think maybe that might be a reason why this behavior is geographically clustered. The other hypothesis that we have is one of influence, where maybe it's not some underlying trait, but the fact that refusal sort of develops in one location, in one county, and then because we socially interact with each other, it spreads from one location to another. So in recent work that we've done, we've basically kind of tried to test this out. So we can design controlled experiments using mathematical models that kind of mimic what these processes, so homophily and influence in this case, look like on different populations. And it's basically the equivalent of having sort of a wet lab setting, but within a computer, right, or a field setting where we can really do controlled systematic studies that allow us to think about how different features affect things. And so by doing this work, we were really able to show that both homophily and influence really can generate this kind of geographic clustering in vaccine refusal, but that one of them, homophily, is actually much more efficient at doing this, especially in populations that aren't particularly spatial in their social structure. Besides doing this in a theoretical setting, we also wanted to do it empirically. So what we then did is we found a lot of sort of large-scale data on socioeconomic traits, which is easy enough because that's collected by the US Census. But then we also, on top of that, found data on social connectivity between US counties. So through social media is one of the biggest ways we all interact with each other, even though we don't live close to each other. And so that really kind of helps us test the influence hypothesis. So based on this empirical data to kind of parameterize these models, we were able to find that kind of how homophily and influence, how these processes can really predict the observed distribution of vaccine refusal behavior that we see in real data. And that's, really kind of confirms that this model is the right one to think about, but also gives us a way to then predict what refusal might look like going forward and have ways to mitigate the threats of that. So I can take a quick pause here. I have about 10 more minutes, so I'll run through next kind of work that we're thinking about on using big data sources for disease spread. I'm going to take a quick drink of water here, though. So in terms of then moving kind of beyond thinking about big data sources for measuring behavior, we're also, of course, trying to use big data sources to think about measuring disease. So again, before COVID times, measurement of disease, so disease surveillance, kind of where disease cases were happening and when, was quite coarse in the US public health system. So that really kind of encouraged us to get creative with finding other data sources. And so one of the main data sources that my group works with is administrative health data coming from medical claims, from insurance claims. So whenever any of us, or 91% of the country that has insurance, when we visit a physician, right, that physician-patient interaction leads to an insurance claim because that physician needs to be paid. And alongside that claim form, right, comes diagnoses and treatments that the physician kind of provided. And that really leaves a sort of paper trail of sort of high resolution data on where disease cases are happening or where treatments are being distributed. And it's, really kind of high coverage because insurance is, thanks to the Affordable Health Care Act, 91% of the US has insurance coverage. And so it really gives us high coverage information in a timely and often also demographic specific manner. But alongside the opportunities that are provided by such data, there's also sort of limitations. So really what we get is, an observation of a particular type of individual, maybe a person of a given age and a given gender that has been diagnosed with a particular disease. But to be able to arrive at that observation, we need to, start with symptomatic cases. So there needs to be a population of individuals that experience symptoms for a given disease. They need to decide to seek care, which only a portion of those symptomatic cases will do. They need to have the ability to access care and the ability to pay. So they need to have access to insurance, which further dwindles that population. That physician that they choose to visit needs to participate in the data source that we have in this insurance claims database that we have. And then finally, that individual needs to be diagnosed and then reported by the patient. So really, just because we're observing this one individual doesn't mean that we're, seeing everybody. It's really a tip of the iceberg situation, and there's a lot more cases underneath. So when we think about repurposing this data for epidemiological purposes, really we need to think a little bit more broadly and think about how to work with that data. And so the way we tend to do that is we For example, trying to understand how, again, this particular disease is distributed around the country, we would first incorporate a disease model, something like a mechanistic model, like an SIR model that some of you will have heard of over the past few years, which really encapsulates the disease transmission process. And from that emerges the classic kind of epidemic curves that we've all gotten used to seeing. Second, we would want to incorporate a lot of covariate information that captures the various demographic and social and environmental and behavioral processes. A lot of the behaviors that I just talked about, right, would kind of go into this piece to explain the variability in disease. And then lastly, because we have to be really careful about the fact that this is not a data source that's been collected for epidemiological purposes, we really have to incorporate a lot of measurement covariates into such a statistical model to account for the fact that we might not be seeing everybody. And so what this allows us to do is think about, first of all, just creating a more accurate map of where a disease, and this is a example with influenza, where a disease like influenza might be occurring, right? Where the left map is kind of what we're observing in this medical claims data base, whereas the right is the model fitted version, right? So it's corrected for measurement biases, it's corrected for the effect that social and behavioral and economic biases sort of factors have on it. And there's a ability to then also fill in gaps, impute data where we don't actually have it. Second, we can then use this to understand which factors that we put in actually matter, right? So all of these factors that we put in, social, political, economic, right, are really just hypotheses. And so the model can actually inform us about which ones truly matter. And then once we have one of these maps, we can really do experiments and think about, okay, how does surveillance need to happen going forward? If we don't have this data source, how can we use this to kind of inform traditional surveillance design? Secondly, we can also use it to understand things like healthcare disparities, right? So the COVID pandemic has really kind of sounded the alarm on the incredible inequities that we have in the US public health system, in particular, in terms of who bears the brunt of disease burden, and then also hospitalization, death, and long-term consequences of disease. And it's really, stark, especially along racial and ethnic lines. And so using an approach like this and using such data, we can really also look at, mapping out where the disparities are. Again, traditional data sources often don't allow us to do this because they're just not fine scale enough. So we can really produce maps that really allow us to do this, and then also use it to understand which factors actually drive these disparities, whether it's the lack of paid leave amongst certain socioeconomic groups, or is it access to vaccination, or is it access to just hospitals and other healthcare, right? Really thinking about this mechanistically can really help us think this through. The last example I'll go to is kind of syndromic surveillance, and this is something we're trying to think about now. So traditional surveillance that we've had kind of through the COVID pandemic, right, really depends on lab confirmation. So all of us at some point during the last 3 years, many times usually, right, has gotten lab confirmation of a COVID positive or negative, right? This is either done with PCR testing or now in more recent years with at-home testing. An alternative way to do public health surveillance is what's called syndromic surveillance, where instead of actually doing a diagnostic test, you track just symptoms based on a symptom profile rather than doing those lab confirmations. And it can just be efficient and timely. And honestly, this is actually how we do seasonal influenza surveillance in the US. But what's really challenging about this, even in the case of influenza, which we've been working with for a long time, we really need to be careful about how we define a syndromic profile. So what we are working on here is, because we have access in this healthcare data base and the insurance claims database to individual level sort of symptom information, we're using that to kind of build a machine machine learning model or logistic regression classification model to kind of identify which symptoms are actually most predictive of COVID infection, since in the case of COVID, we did have a lot of lab confirmations. And results from that model look, something like this, where various symptoms kind of feed into our prediction of who might have COVID. And that allows us to, sort of predict at the individual level, who might have COVID, and then scale that up to the population scale. What this is opening up an opportunity to do is really allow us to kind of supplement disease surveillance going forward, even for pandemics, but certainly for endemic infections, things like seasonal flu. So what we're looking at here, the orange curve is what was measured for COVID in spring and summer and fall 2020 from PCR testing. Mostly at the time, there was really no rapid testing at the time, so mostly PCR testing, whereas the blue is maybe what we might infer based on a syndromic surveillance system designed based on these insurance claims and the symptom profile. And so, we see lots of cases of what looks like COVID, but really maybe early on, well before March 2020, right? Maybe that's just the flu season's background noise that we're seeing. Whereas, much later on, where we have both lab confirmed data and COVID syndromic surveillance COVID data, we can see that the syndromic surveillance COVID data really matches what we're seeing in the lab confirmed data, but it picks up a lot more cases that are not getting tested. And it also maybe allows us to track COVID in that March to May period where really there was no testing happening, but we'd still really like to be able to measure what COVID looked like during that time. So this is kind of just ongoing work at the moment that we're doing, and stay tuned. If this result stands, we'll be really in a much better situation in future outbreaks. So I'll finish up here with some just kind of parting thoughts, right? Really from a infectious disease perspective, what I'm really, my group's really working on is thinking about both individual level behavioral heterogeneity, which is relevant to kind of explaining variation in transmission, both in terms of disease outcomes, but also in terms of like what outcomes we're seeing in terms of downstream effects of COVID, but then also thinking beyond the individual level at this landscape scale to think about, it's not really just the sum of its parts that matters when it comes to this population scale or landscape scale. Really, there can be heterogeneity that arises there, like in the case of the spatial clustering with vaccine refusal, that's independent of just the individual level processes and more than the sum of its parts. In terms of data and modeling, really what we've been trying to do, and we really think this is a important way forward, is pairing models with data, right? So we can't really do any of our work in the biomedical community without data, but really thinking about both statistical models as well as mechanistic models really is the way forward to kind of produce a strong mechanistic understanding of these very complex systems. When it comes to sort of novel electronic data sources, I really think they provide us with a ton of opportunity in this field. But I would say let's proceed with caution. So I'll wrap up there and give a huge shout out to my amazing group, both recent and current members, who make it possible to do what we do, and an amazing group of collaborators, and grateful for funding support as well. So thanks so much, and back over to you, Jack.
Shweta, thank you so much. I was really excited to see your kind of use of those non-traditional data sets at times and leveraging those. And I think that's an important thing for students to appreciate. I know a lot of students kind of will, like, for example, they say, well, this isn't like, COVID data or something, but it's like around COVID, right? It's peripheral to it. It's the behaviors, for example, or it's those sociological factors, which would, COVID would influence. And if you look at those, you might get some insights into the behavior of the virus or the disease trends or what have you. And I was thinking it as you were talking about how my kids are older now, but I remember when my wife first thought she was pregnant, she started looking up things online. And of course, what happens, right? Well, somebody's out there watching, and then all of a sudden you start getting all sorts of flyers in the mail, right? About,
Right.
oh, diapers and formula. And all of a sudden it was like, all she did was like look up, pregnancy symptomatology. And now and we see this all the time now, right? But it's
Right.
these behaviors. Somebody did something and it influenced something else, and it kind of has this little knock-on effect. And that you're noticing that there are mechanistic relationships between these things is super important. And I think that's probably something for students to appreciate too. And I'm wondering if you might comment on that.
Absolutely. Absolutely. Right. So really, in some senses, we are, as scientists, coming late to the game, because the tech industry and marketing folks, right, have been using these data, these kind of, digital trails that we're all leaving behind as we use the internet, right? They've been using that for years. This is how, you all ended up with, flyers at your home, and we all experience this in our inboxes and so forth as well, right? And so really, I think it's high time, and of course, we're not the only ones doing this, but it's really high time for us to be thinking about, in the scientific community, okay, if we, of course, study design and classical scientific studies are, the need for those is never going to end, right? But because we can be limited to how large we can do those studies, or for, so not just logistical limitations, we can also have ethical limitations. So in those contexts, can we leverage the data sources that are just out there to kind of make sense of these complicated questions is really what this is about. But yes, I do think so and not just from students, I do hear this from scientific colleagues, science colleagues as well, where they'll say, okay, well, this isn't perfect, right? This isn't, and in science, we're often taught to like design the perfect study. And this is always going to be far from perfect. But I think what's important to remember is that we now have a lot more tools, especially with statistical modeling, that really allow us to carefully use these data while accounting for biases and gaps, and still try to generate new understanding, at the least generate new hypotheses that can then be addressed using traditional approaches, field studies or wet lab studies, and so it seems like an opportunity not worth missing to me.
Are there any opportunities to connect with, say, the consumer industry about, looking at these things? You kind of like, for example, the behavior of, going to buy cold medicine or cough syrup or something, and like, oh, hey, look, there's been a run on cough syrup at all the local CVS's in, Nashville, Tennessee. And you go, I wonder, is there an opportunity to partner with some key stakeholders like that?
Absolutely. And really a lot of the work we've been able to do over the last 3 years, and a lot of the data sets that have appeared during the pandemic, right? They're not new data sets that suddenly started being collected during the pandemic. Some are, but really the reason, for the data revolution, as I call it, during the pandemic, is because those data were being collected often by sort of the consumer and marketing and business community. So that data set that I mentioned, that's based on like app usage, location tracking, where, it's counting the visits that I take to the CVS and then to the grocery store and then home, right? Those are, usually measured for marketing purposes. And so really the release of those data to the scientific community is what's made some of these studies possible. And it's let us answer so many more questions about kind of how we move around and what behaviors we engage in. So just that's kind of the bigger scale version. And then, there's been more specific tracking of, particular product use, or even like Google searches. This is probably the oldest version of this, of looking at Google searches for just signals of what people are thinking about and what people are doing. That really started all the way back in 2004, actually, well before the pandemic. But it also, did have a massive failure at some point, and it's because the need for kind of a careful mechanistic understanding and the need for carefully accounting for biases hadn't been appreciated. And so really what at the time they did was they just took these data from, let's say, Google searches and fed them through a machine learning model. And of course, the machine learning model worked great while the data looked, while reality looked like the data, and then like the training data. And when it didn't, it broke quite severely. And so I do think we've learned a lot since that time. And I think it's important to just, move forward with caution.
You talked about early on, and you gave some examples of mechanistic modeling. Can you expound on that? What does that mean? How is that different than just say, getting some data and stuffing it through your favorite machine learning algorithm?
Yes, that's a great question. I wish I had actually had a chance to include that slide today. So really, right, the field of infectious disease does tons of quantitative work, and I tend to break down that landscape into two groups. One is what I would call phenomenological models, and the other is mechanistic models. So phenomenological model is really where it might be a machine learning model where there's no sort of underlying biological or social processes or mechanisms that are included. And really our goal is to make the model predictions look like the data, right? And so if you maybe look at disease data for COVID early on, right, it seems to go up and seems to come down. That's a classic epidemic curve, right? But really all those models would do is just be able to predict that shape without any understanding of what the underlying processes were. In contrast, a mechanistic model would be able to predict that shape based on just underlying processes. So in the case of a disease transmission model, a disease dynamics model, what you would feed into the model is not what's happening at the population scale, even though that's what you're measuring. You might be measuring the number of COVID cases in a community. That's not what you would put into the model. What you would put into the model is the interaction that needs to happen at the individual scale for a susceptible individual to be infected by an infected individual, right? That is the fundamental disease transmission process. And that process is what you would bake into the model. And then the model would be responsible for taking all those individual level interactions and predicting what might be happening at the population scale. And it turns out, after a couple of centuries of this kind of work, maybe more, that if you just bake in a simple interaction process, a disease transmission process at the individual level, you're actually able to emerge from that epidemic curve that we see in reality. And so that's an example of a mechanistic modeling process, and SIR models or compartmental models, if you all have come across those over the last few years, right, are an example of that, where they're really built on the underlying social or biological process that's relevant for this system. And they don't rely simply on the data. I won't say the data doesn't matter, but they don't rely simply on the data. And so they tend to be more robust and reliable for making predictions, because if, again, future data doesn't look like past data, then those phenomenological models have nothing to fall back on, whereas the mechanistic models still have at their core this basic understanding of mechanism, and so they can still make predictions. They also have their limitations, so let me not pretend that that's the case. That isn't the case, but they're just philosophically quite different.
What final advice would you have for people who are interested in this topic, interested in getting into this? They're either, people who are data scientists or people who are like, epidemiologists, for example. What advice would you give them if they wanted to take on some of these particular challenges like what you described?
Of course. So I invite kind of everyone to join us, because we are dealing with a mess of problems, as the field has demonstrated over the last 10 years, and especially we really need a social science perspective. So if that's a perspective you bring, please come join us, right? Behavior is at the core of this, as is, the interaction of infection decision, public health policy decision making with political processes. So really, we do need a social science perspective, but data science, right? Skills and perspectives are absolutely needed as well. Honestly, I wouldn't have said that a decade ago because we just were a fairly data poor field. Again, mechanistic modeling and the theoretical work really kind of took us very far up until that point, but the last 3 years have really opened up the amount of data out there. So yeah, would love to have folks join us that bring that data perspective. And I would highly recommend also kind of learning, if that's not your background, also learning what the mechanistic approach kind of brings to the table, because then I think that pairing of data science with mechanistic modeling really is it's a powerhouse of a combination. So.
Shweta, thank you so much for sharing your work with us. Technical difficulties aside, thanks for handling that with such smoothness. But thank you so much, because this is such an important area, and I love the idea of leveraging these non-traditional data sources, basically non-direct, right? There may be more indirect data sources, but they're directly relevant. And thank you for sharing this idea of mechanistic modeling and all your wonderful work today. Thank you.
Thank you so much for having me.
You bet. And everyone have a wonderful weekend.